everybody at least read the blurb on the Meetup. The objective is people to get together and learn, build portfolio of data science work. So there's quite loaded in the sense of portfolio. We can talk a lot more nonstop, but this is the number 4 that we started several weeks back. And ideally, we were hoping that we will get to a stage where we come together and then just build things. Build model, build data product, and build something together. So sort of treat that as, that's why I called it a data club. But before then, we hear feedback from people who come in to wanting to get a bit more of the content that's a little bit structured, because we do have a good portion of people who come here with not a heavy data science background, so therefore they had expressed interest wanting to hear some more structured information and content. And we're just lucky enough that we have people that who can do this, including today's speaker, Ben. If any of you that feel that you could have something to contribute to the group also, it would be fantastic and very low-key, low stress. Hopefully, then you just feel comfortable, talk whatever you know, and stuff like that. So that serves as another invitation also to everybody who wants to talk about some topic that's other people can learn. So with that, my name is Ying Li. I have been a co-organizer with the person sitting there, Kenny. And also we have Bobik. He actually is pretty sick today. He didn't want to come and spread the germ. So the 3 of us have been organizing this set. We've been together doing data science, and we started doing our own hands-on meetup work more than a year ago. Just meet on Saturday every week or every other week together and just build things. So it has been a lot of fun. Therefore, we want to spread that, to get more people and doing that together. Not necessarily to form a team per se to do the Kaggle competition, which is oftentimes the first reaction people think when they say get together to form a team to do Kaggle competition. Not necessarily it's my passion per se. I actually more passionate about building a portfolio, which is actually express lots of your creativity. If you think about artist or designer, the concept of portfolio is what you can do, how you express yourself with different angles, dimensions, tools, and all of that. So anyway, I'll stop here.
As you said, I'm Ken Harrington, and I don't think we need to go through introductions right now. Maybe we should just get started with Ben's talk, and then maybe we can go from there.
Yeah. Now we can just exchange. So if you have questions, just raise hand and talk.
Okay. So I wanted to do a little bit. I probably ended up with more background than I originally intended, and actually I know the data that I picked is actually different than what I described. So, but it's similar, but we'll get there. I also realized how rusty in R I am because I've forgotten. I used to do a lot more, and then for a while I was actually telling analysts what to do. So I realized that I wasn't quite there. I just put my Twitter. If you wanted to, reach me. Otherwise, you can get through Meetup or talk to me about my email after. So I'm going to start out with a little bit of molecular biology at the beginning. I should say I'm a computer scientist, so I know just enough to be trouble. And I actually have written journal articles laying out research directions and things like that, but sometimes I find it funny that happens. So I know enough to explain what you need to know and what you might want to do to figure things out. So I'm going to lay out a little bit of background that helps you understand where the data is, what the data is that we're looking at, and what it has to do with anything. Okay, so we have multiple different kinds of molecules that we're going to look at. The first is DNA, which if you're familiar with programming, is a little bit like a program in the sense that it describes the pieces that go into our into organisms, the construction of organisms. There's RNA, so I'm not going to get into the chemistry of this, but there's RNA, which is constructed from the DNA, which is, for our purposes, it carries information. So sometimes the information is, as we'll see, I stole some videos, which probably will look bad on the video, but the main purpose of RNA for what we're talking about is basically taking the DNA and copying it, or copying parts of the DNA so that we can construct proteins, which is really what the molecules that make things happen in our cells. So within that framework, these molecules fit within what's called the central dogma, which is basically how DNA becomes proteins. And there's all kinds of, the actual central dogma is kind of complicated, more so than people thought originally. I always misremember my slides. I don't remember my slides, but anyway, so I'll get more into that, but we're going to talk about the first step, which is transcription. There's actually audio to this, but I figure with the video, let's not do that. So what's happening is something is causing, the DNA is in this helix form, which you see here, and something has caused it to open up, and these proteins, jumping around, proteins have come and attached to the open DNA. What's going to happen now is that these are going to start attracting other pieces, which are called transcription factors. Well, that's actually, and they build up this ribosome, this compound here, that is sitting there ready to go, and then this thing's going to come, bam, oh, and it's going to open up, and we're off to the races. So what's happened there is this molecular process where we built up this complex that said it's okay to copy this part of the DNA. And then as this thing that came whipping around it doesn't always happen that way, but it's something that attached to a different part of the DNA, came around and actually activated the process, opened things up so that this huge complex could go rushing along and then create the RNA, which is what's coming out. What you see floating around are the little pieces that are going into the construction of the RNA. It's kind of like a stochastic process. There's these things floating around, and they come in, but it's very deterministic. It's actually copying exactly what is on the DNA, the particular strand of the DNA that's there. Yeah, James?
Yeah, I just wanted to say it's not the ribosome, that's the transcription complex. The ribosome is in the translation.
Oh, right. Sorry. Yes.
Just in case anyone's, because you probably meant to say,
It's not going to matter, yeah, to them, but it should matter. I should use the right word. Okay, so that process, it actually is, there's a discrete translation that occurs. The RNA is copied from the DNA. So there's all these things that are going on at the beginning of that. There was this particular spot, there's these spots in the DNA where things happen, where it opens up, and other things can come in and attach. And so at that particular point, there's these particular places that are important on the DNA, and we'll talk about the data that relates to that in a little bit. So that process is the first thing, that string of RNA comes out, and then it actually is going to, that all happens inside the nucleus of the cell. It's going to come out, and then it's actually, there's a step in between. Sorry. So the gene that it was just translated in that picture, or transcribed in that picture, has a place where we saw that dog ear attach at the beginning. It's called the promoter region. And then we have this arrow which indicates, in this case, which way we're going to copy. So once we start transcribing, it just copies everything until it reaches a particular code in the DNA that says stop. So everything is, in that respect, is very deterministic. Now what happens after that is that it depends a little bit on the gene and what's going on. There are different parts of the gene. So the gene actually has, so what we've got the promoter, then the stuff that's translated is the red and the black. And so we get two parts. The red, these exons, are the things that actually are used to create the proteins. The introns are things that are not. So they basically they're pieces that are, they probably serve some other purpose. They get actually sucked out of, they either serve some other purpose or none at all. They get cut out of the RNA, which then, and then the RNA gets put back together, okay, into this, the processing stage. So what can happen is that we can skip, we can actually put these things together in different orders and different genes, what we might think of as this thing here, could be put together in different ways. So each of those different ways might constitute a different protein, or give us a different protein in the end. Okay, so this thing is what we call messenger RNA, what results here. And then that thing's going to go out of the cell. All right. So here comes the RNA, or out into the cell from the nucleus. So it's coming out of the nucleus. I like how these things look like snakes. It comes out into the, just floating around in the cell, which is really densely packed with stuff, but it's easier to show it this way. And what happens now, this is the ribosome. So the ribosome is what's actually going to construct the protein. What it's doing is taking the pieces of the protein that are attached to these complexes here, these green things, and it's essentially reading the RNA, the messenger RNA, it's looking at chunks of 3 nucleotides that match up with this complex, and then it attaches the amino acid that's on the complex to the protein that's getting constructed. So these things come in, they basically read it's like reading off the RNA, and it's a machine, it's constructing, it's a molecular machine, it's constructing this protein from the code that's there. All right, I don't remember what that step's supposed to be about. But that's basically, that's what's happening, is that we have these steps where we have these molecules that go through and that first copy the DNA into this messenger RNA form, which then gets rearranged. And then we can, once it comes out of the RNA comes out of the nucleus, then the protein is constructed. The thing about proteins is that they have, one of the things to be aware of about proteins is that they have structure, so 3D structure. DNA, we know, has this helix structure that we saw in that picture. That has more, I don't know exactly where that comes into sort of how the molecules fit together, but with the protein, it actually, the purpose of the structure has to do with the function of the protein. So the protein, understanding actually how the protein goes from being the string of amino acids to this blob of stuff that then has certain parts of it exposed and certain parts of it not, or has little niches or places where things come in and get modified. All of that is very complicated. So there's a lot of people that's what they do. They try to understand how do you take it's really easy once we know the gene, we can actually figure out what the protein, the string of amino acids is going to look like for a protein. We can do that. We can easily write programs to do that. But the problem is figuring out what happens to what the protein actually looks like once it folds up. So that's actually a big computational challenge that people work on. Not so much one that's like a data science one, but it is important to understanding how things work. Okay, so we have actually multiple stages where things are going on. We've got the transcription. We get some modification that happens there. We've got translation that gives us the protein sequence. And we might have modifications at this point. And then we get folding that gives us the protein with the post-translational modifications. Other things might attach to the protein. So the central dogma is all about just how we go from the DNA to that thing at the end, the protein at the end that actually does the work in the cell. Okay, so what I wanted to do was give you a brief introduction and then talk about the generality of genomic data, and then we can look at our problem. Okay, so this is a site called the Genome Browser at UCSC, and it deals with what's called genomic data. And so I wanted to sort of give you an idea of the variety of genomic data that's available for what we deal with, just so you can understand the different kinds of things we have choices about once we go. So the website is just genome.ucsc.edu. And it actually has a couple different things that are useful here. I don't use this so much. I use what's called the table browser, which allows me to get data out of it. But this actually shows you what's in the tables. So this was a protein. This is actually a gene that is actually related to a project that I can't seem to shake off. I'm hoping it's going to be gone this week, but it still keeps coming back. And let's look at what we have here. This is part of the genome. It's one of the chromosomes, chromosome 4. And the chromosome has structure. You've probably seen the pictures of karyotypes. They look like, you kind of see like these X's, the pieces, and in the middle where it's kind of crossed over, there's this, that's the spot called the centromere. So that relates to that structure. And then these bands actually go back to that when people were dyeing things dyeing the chromosomes with dye and taking pictures of them. Those are actually the bands that you would see in the pictures. And people still talk about those things. Cytobands. Although usually they don't come into play a lot with the stuff that I do. So in this particular case, what this is doing is it's just kind of giving you a picture, if you're familiar with that sort of thing, of where this particular gene is located. The region we're looking at is on this particular chromosome, chromosome 4. We've got the location. We know how big in terms of how many nucleotides we've got, how big this region is that we're looking at. And we're focused in particular on this one particular gene here. So what I've got here is that, remember I had the picture in the central dogma, it had the red pieces, the red chunks. what we see in this picture is actually these black squares. Those are the red chunks. So in comparison to that picture, this is the pieces of the pieces of DNA that are actually contributing to the gene are really really far apart. There's longer chunk here, but the rest is these separate chunks. Now you'll notice all of these things are, it's listed a lot of different times. And the reason is that we have different ways of identifying genes. Different people have different ways of identifying genes, and they don't always agree. So we have, this is what UCSC, University of California, Santa Cruz, calls a set of genes, which is a compilation of all these things, which are different ways of looking at what's a gene. And so one of the first things when we're talking about genes, when we want to talk about what is a gene, which genes are we talking about, we have to decide whose model are we going to use. And so there are actually, you see multiple there. I'm not going to go into detail about it. I just wanted to point that out, that that's one of the things that you have to consider when you're talking about this. Now sometimes this RefSeq, it's called reference sequence, what it will show you is actually, so this is one set of models, which is good. We like to have just one. It will show you alternative versions of the gene in terms of how it's spliced together, what pieces are put together. So you can see this one is different than the others. It ends in a different place. Usually what will happen is that the gene will start in a different place, or it might drop out some chunks. Okay, so all of this is about, these are about the genes. This is about the messenger RNAs that have been identified in that region. And it just keeps going. I don't want to go through all of these things, but it's got information. There's information about places where the genome, where the chromosome are not, where the chromosome will open up, is prone to opening up. So these DNA clusters there, this part here, actually has to do with that. It's recognizing a property of the chromosome where these things will happen. It also has places where they've identified transcription factors. Those places where the dog ears had attached. And then more interesting to me because of the work that I do, all of these things are places where there are changes that commonly occur on the genome. So just in this one gene, all of these things, and we haven't even gotten to the bottom yet. So these are all changes that might be present. Well, they're all places where mutations, or not mutations, they're all places where variations occur. So it just keeps going on and on and on and on. All right, so that shows you just the genomic data. The other data, there's a lot more different kinds of data. There's other kinds of data that we might encounter. So that's describing the gene region. Yeah?
You said this was chromosome 4?
Was that chromosome 4?
At a high level, do you know what chromosome 4 is responsible for?
Well, I mean,
Many, many things, of course, but
it's, yeah, I mean, genes, what happens is proteins actually do things, right? So you would be, how many things on chromosome 4 do you want to pick out? I don't know off the top of my head.
Sure.
I have a question, Ben. So
Yeah.
the data that you are showing, it is the distance, or so like how it is being measured? It looks to me that these are very microscopic.
They're very small, so let me see if I can get back to that. It's not
Then,
opening.
so the question is that what is this data, and secondly, how it is being measured?
Okay, so this is all of this is describing a region on the genome. So basically, it is it describes part of the actually something I left out. So I should go back. So it's part of chromosome 4. We know that it's this region right here, from this position, counting from DNA is kind of two-ended, so we have the chromosomes are two-ended. So we can start counting from there, and going this way, or we can start counting from here and go that way. Genes can be on. There's two strands together, and they go in opposite directions. So what this is actually showing is a region of that. So if we just focused in on the region of the chromosome, and we're actually looking at this region of thousands of bases, thousands of individual nucleotides of the DNA. All right. So all of this is based on it's just showing. It's like a map, basically.
And how it is being captured?
So it depends on what it is you're talking about. These things, the way that we would figure out a gene now is we would use sequencing. And that's what they were doing before. They were recognizing features in the sequence. So basically, the sequencing occurs. They take the DNA, they essentially cut it up into pieces, and then they have to reassemble it.
Okay.
All right. And so as they go, there are things that show up that indicate that there are basically chemical or molecular markers that indicate where these boundaries are. And you can do with the sequencing, with next generation sequencing, you can actually see it happening in the data, but it's a little harder with the older style.
It would be probably safe to say you can think of it as just a long chain of 4 characters, really, really long, like millions and billions. And occasionally a couple characters will be different from me and you in this particular region.
Okay.
And that's what Ben's going.
Well, each one of these regions is each one of these parts of sets of data is determined in a different way. So I can't give you a short answer to that question.
I think I have to do my reading.
Yeah. So I will point you to the NCBI site, the link. I already did actually in the Meetup announcement. So there's a lot of good tutorial information there. They actually have some books, some free textbooks online. Now the one thing I did want to mention, and sort of when I was talking about the assembly, this is how they got these sequences in the first place, has to do with that business at the very top there. So this GRCH37, HG19 business has to do with that's a version number, or those are different version numbers, they just happen to be different ways of identifying it. So that 2 different groups were working on the genome, human genome, and then they came together, and so they have different nomenclatures for the number. So it's human genome 19. So this is the one of the recent ones, and then they'll have version numbers after that. But I can't remember what the version numbers represent. They don't. What can happen between 18 and 19, genes got up and changed neighborhoods. So you'd think a gene was here at this particular. So this gene might have been in a completely different place on chromosome 4 in assembly 18. All right. So it's really important when you're talking position that you know what coordinate system you're working on, because things can change. Now, what wouldn't necessarily have happened is, so we know that based on all that stuff that we saw before, there's actually a chunk of DNA in front of this beginning that is important to starting things up. So if we're interested in stuff that's happening over here, generally what happens is that if this gene changed neighborhoods between assemblies, that stuff went too. So if you, it's not always the case, but you can have stuff move.
Now, Ben,
Yeah.
is it safe to say that a gene corresponds to a protein?
So a gene is generally one-to-one, but a lot of times you can find genes that have multiple different proteins that correspond to them.
Sure.
So usually, like these RefSeqs, actually, the RefSeq genes, the RefSeq identifiers usually correspond to what we call a transcript, which means that that's the thing that's going to get turned into a protein. So it might be that they will be different, right? And they'll actually have different numbers that go with them. I didn't get into any of that nonsense in my stuff today. And actually, I'm showing you human stuff, and we're going to do mouse. So I'm not sure I've explained that, but it had to do with some history that I got into. I was like, oh yeah, I worked on this data a long time with students a long time ago, and then I got lost in it. Okay, so the work that I've done is actually trying to understand genetic disease. So I want to talk about that kind of in the context of understanding a little bit about what data we use and why we use it. So the idea in genetics is that we are studying something called a phenotype. The phenotype is the thing that we see. It's whatever, if we're talking about plants, it might be, corn that's yellow instead of white. It might be a dog that is brindle, the little stripes, kind of like tiger stripe, but as opposed to spotted. And it can be all kinds of, it could be having a disease or not having a disease. And so this is the thing that ultimately we're interested in explaining. And it comes in that the idea behind genetics is that we have two things that are contributing to it. One is genetics, and then the other thing is what we call environment. So when I started working on this, environment was kind of vague and funny, and people weren't really sure what it was about. But really what it turns out is that there are things that happen that cause modifications to proteins or to your genome, actually, that are not really changes in the sense of what we typically think of as variations, like those little slashes we saw on the website. Those are actually spots on the genome that are different. So if we took Kenny and myself, there's going to be a lot of different places that we have single nucleotides that are actually different from one another. Whether or not that makes any difference or not is another thing. But there may be other spots where somehow our DNA is modified in different ways, in different biochemical ways, that not changing the nucleotides, but attaching other complexes or something, that are caused by the fact that that I used to explore caves and was exposed to bat feces or something like that. So that could somehow change my
Phenotype.
my genome or whatever. And it might be that something that you've gone through has modified it in other ways. Those things can also result in there might be other situations where, for instance, sort of a classic story with of people with bipolar disorder is that they can go without being diagnosed for years as children, and then they go off to college, their introduced into a stressful environment, and then bang. All right, so one of the, they're dancing on the table. All right, and so it may be that there are triggers that are coming from the stress that are modifying something, it's kind of like in that picture, it's not exactly the same thing, but in the picture we saw the thing kind of come whipping around and cause the chemical reactions to start. So these kinds of things can come from the environment, can affect us in other different ways. We're really just starting to understand that in much better than we used to. So there's variations of all sorts that are leading to the outcome. All right, so this is actually a picture. So I've done a lot of work with genetic diseases in kidney. And this is a picture that I drew for a survey paper. We have a lot of different things that lead up, that are kind of like machines that lead up to us having things that we can observe as either through clinical measures, actually measured in your blood or your urine or whatever, that relate to our disease in the end. So I'm in this particular, I stole this picture from the kidney disease. But what happens is that how we get to that point could come from variations in that affect multiple different parts of that system, cellular system. In this case, this is the cellular system. We also have tissue, everything kind of combines to build up tissues and how things fit together. So we have genetic variants. They might actually, so I'm interested in these changes to the genes. Some of them can actually change how the protein, those dog ears attach, whether they attach or not, whether that thing comes around and smacks it to start the process. It actually can, the most common, or one of the most common genetic changes is actually something that causes the protein not to work. So there have to be particular nucleotides in it, 3 particular nucleotides in a row for an amino acid to occur in a protein. If you get the wrong nucleotide, you will get the wrong amino acid, and the protein could be completely wrong. So it won't fold up in the right way. It may, may be reabsorbed or whatever by the cell. So there are all kinds of things that can happen, and that's actually where a lot of simple genetic diseases come from, these sort of changes that occur immediately to the protein that change it directly. So we can have those changes, and they're affecting, we know that they can affect, like those could affect the regulation, which genes get copied. This protein machine, how the proteins interact with each other, that can be acted directly either through directly based on changes to which the protein, it might be that the 2 proteins can't work together anymore, or they go through this regulatory machine that says proteins don't get produced at the level that they would otherwise. So it can be subtle kinds of things. All of that accumulates and results in what shows up at the bottom. But the problem is that when we study these diseases, so now I'm getting into my own little biases here, but when we study these diseases, we kind of study everything together. We're trying to aggregate it and see the results of everything and how it's coming out here. What I think is happening is that we have, the reason I've got these little pages here is that there are different groups of these changes, whatever they happen to be, that are working together in different people. So I could be here having diabetes because of a particular set of changes there. Somebody else could be having diabetes because of another set of changes. They all come together in different ways. And sort of the analysis that we've been doing is not focused on that kind of thing. And that's actually the work that I do when nobody's looking. That's when I'm trying to figure out how to analyze. Okay, so what we try to do when we're looking at this is we can look at different kinds of data. So the genotype, that's the technical word for the figuring out what genetic variations we've got. Right? So which of those nucleotides are changed into which things. So all the changes at the top. We have the regulatory, everything else. So those are measures, are actually identifications of, is the genome changed in a particular location? Then the rest, these are actually measures of abundance. So literally what they are is we're trying to find out how much of the stuff is there. So gene expression, we're actually measuring how much messenger RNA there is floating around in the cell. So as far as measuring, what happens is the cells get smacked, their guts spill out, and we isolate the RNA through a lot of steps that fortunately I don't have to do. And then that goes through a molecular a process where we actually measure how much of each messenger RNA there is. And I'll actually talk about that. The proteomic, that's measuring how much proteins we've got, and the metabolomic is measuring how much of each metabolite we've got. Now, the issue is that for us, the issue is I want to talk about publicly available data because that's what we can do stuff with. There are some data sets that are protected, so you have to get permission basically in order to, you have to be able to register for access. So not all of it's publicly available, some of it because there's concern that it could be used to discriminate against people because of health risks. So if you're known to have a genetic factor that is believed to lead to some chronic illness, then the concern is that employers could use that or health insurers could use that against you. All right, so in this case, those data sets are protected. But there is a data repository for those kinds of things. So when people do large studies, they bring in the data. It actually is supposed to go into these repositories so that people can have access to it. It's just there's hurdles to go through to do it. Some things, like some of the data that I've worked with was from Native Americans, and that data has extra levels of protection just because of the history that medical studies have had with people in general, indigenous populations, and even African Americans, for instance. Okay, so I left the bottom. So there's reasons that we can't get a hold of these. These things generally aren't available because there are either so many things to look at. It's interesting that they're really not easy to collect. There are high throughput ways of doing it, but it's just sort of really awkward. They have these really big machines, mass spec machines that measure abundance, but it's basically what they're doing is my understanding is they kind of essentially spit the proteins across a chamber at high speed and measure where they hit. Or there's all kinds of different ways that they measure. But they're not as high throughput as the gene expression or even the genotype. Metabolites, again, so these are different, and part of it is we don't really know exactly what proteins are there. It's like you would think we would know, but we don't. So we don't actually know all of the proteins. And in the kidney realm, they're still worried that they don't actually know what are all the things they want to look at. But gene expression, that's one of the most common forms of data that's available, and it's right out on the web. You can get it. The thing about genotype, actually the reason that genotype is hidden is that if you have enough information, you can figure out who's in a study. So there's there's ways to do it do it statistically. So it's kind of this statistical. It's basically the same concerns that you would have in a statistical database in uncovering private information. Sort of the same kind of principle. Okay.
Ben,
Yeah.
quickly, that sounds very counterintuitive, right? Because, like, how could we re-identify the person, actually?
You already have to have some information, genetic information about them. There are ways to do that. There are ways that people could do that.
Like, can we just take the person's name away?
Oh, no.
Okay.
Yeah, you would have to have some genetic information from them already.
Or their parents or their family.
Yeah.
Otherwise, the study is rendered useless, or what?
No. It's the concern is the exposure, right? So the concern is that somebody could have enough information that they could figure out that Kenny had some disease that I don't.
Like, well, why not just make it available, not take away all the identification information and make the data available?
Oh, so you can actually, the studies are set up so you're involved in a study, you would be identified, your genetic information would be identified as being a subject in a study of bipolar disorder. You would have no identifying information.
Right.
You would just the genetic information.
Right.
And so you would be either a case or a control in those situations. And if somebody had enough genetic information, they could figure out that you were a case.
Really?
Mm-hmm.
Wow. But
I think it's not. I don't think it's a really big deal, but for some people it is.
But really, if someone with the enough data, they can really pinpoint to me, it sounds like they have already solved the entire disease identification problem then. Right?
No,
if you can solve that kind of
they can
equation,
figure out whose sequence, if they have enough of your genetic information already, that they can use that to match against the information from the study and figure out whether you were involved.
Okay.
Yeah. So it's so right now this is something that came up, I don't know, maybe 5-6 years ago. Somebody identified there used to be these data sets were wide open, and then they just somebody figured out that you could do this, and they just closed
because
it down.
if, like I come from the internet data background, right? If you have the massive click streams,
Right.
you can reasonably figure out who that is, because people tend to search their own address, people tend to search their own name, their employer, their friends, right?
Yeah, I think we're in a different situation.
So like, how could
I
this
think,
be identified?
Yeah, I don't think this is. I honestly don't think this is any worse than. I mean, you can figure out probably more stuff from what searches you do than this, but so. So it's just that's the situation. The data is still clamped down. We can't get a hold of it. Now there are projects like through ISB, the Institute for Systems Biology, where people are their genome is being completely sequenced and it's available. It's a limited number of people, and actually some of those people are walking around some of them are giving talks and saying, I used this data from this study to identify that I had this particular disease, and I walked into the doctor and I said, I have this disease, and they said, you're crazy, and then they went through all the. So there's already people that are using the data, and they're very open with it. So there will be data, but it's just not as abundant as the gene expression.
Ben, one quick question for you. The direct-to-consumer DNA studies like 23andMe, where do they fit on this? It's just gene expression, or is it genotype?
That's the genotype.
Okay.
Yeah. So you take a swab, mail it off to them. They tell you actually what the genotype is. So what they had before was they would have before they got in trouble with the FDA, they had the genotype, and then they had the information about what genotypes match up with diseases have been found to match up with diseases. And then, so since that kind of leads to, I have these genetic changes, do I actually have the disease? Oh my God, I have the disease, I'll cut off my breast or something, right? So, that's why that was closed down. I guess there's basically what happened is that now if you have that information, you will go to another website where they have curated the information about which genetic or which variations relate to what diseases. Yeah.
Yeah, so the concern with 23andMe was that the way people were behaving with the data, or was it also that the conclusions that were drawn weren't always correct? Was their analysis correct?
So the issue, yeah, so there was, in terms of conveying the information, there was nothing wrong with it. The issue was that it kind of borders on the diagnostic. So if you were to go to a hospital and have a genetic test done, they don't do these panels. They don't do the whole thing. They would have some reason to believe that you have a particular disease, and then they would look for particular markers. And in that case, what they would do is they would look for those things, and then if they found them, or if they didn't find them, you would sit down with a genetic counselor. So there would actually be a person that talks you through how to interpret them. What happens with 23andMe is that you get this report. It could tell you all kinds of things, and told lots of people all kinds of things about genetic risks that they had. But they're from studies. So the studies that I've dealt with, and they don't really tell you for sure anything. So the answer is that basically there is no, so if you came up with these set of risks that you had no guidance in terms of how to interpret it and how to deal with it in your own situation. And so from the standpoint of the FDA, they were kind of stepping into the bounds beyond just providing information into something that was medical, because people might make medical decisions based on it. So, and again, it's all of this stuff is, I think eventually things will change. But right now, actually, if you had the 23andMe report that told you all those risk things and took it to your doctor, they would have had no idea what to do with it. Most of them, until recently, most doctors were not educated in thinking about genetic diseases. Okay, so I want to narrow down on gene expression technology. And I'm just going to show you I got a video for this one. I don't know if it's any good or not, with the sound off. But there's two different kinds of ways of identifying what genes are expressed. What they do actually is there are, the DNA is being copied onto messenger RNA. The messenger RNA then comes out, or is floating around the cell, and it's basically just kind of out there, right? So it's available to be translated into a protein, but it's just kind of floating. And so basically what we do is kind of open up the cell and check to see if it's there. What we can do is attach, if the RNA will match up with DNA that's complementary to it, so the cDNA. And what we're trying to do is we're trying to find these short fragments of DNA that tell us which things are showing up, which genes are showing up as being produced. Right? So they're different, particularly designed probes of DNA. And then we tag them with something that, if we shine the right intensity or right frequency of light on it, it'll light up. So that's the classic thing, and it's based on this idea of designing these probes. The other thing that people do now is because we have these sequencing, high throughput sequencing machines, they do RNA-seq, which basically is identifying these RNA sequences that are occurring. And so then in that particular case, we're actually recognizing the sequence as it occurs. We're actually reading, it literally reads the sequence although they're read in fragments. Okay, so we have this problem where we might identify an RNA, but then we have to pull it together. The first one, we're actually choosing fragments that are, at least in theory, are going to identify the mRNA that shows up. This one, we're actually looking for the mRNA. So there's a lot there we could go into, but this, I don't know that this is a great video, but we'll see. Okay, so this is actually a microarray chip. On this, there's actually all these little spots. So little teeny spots. Each one of them has bound to it these probes, or cDNAs. Yeah, it's not very useful without the sounds in. Okay, and there we go. So what happens is that as the sequences get bound, when we look at them, the intensity or the color, depending on the technology, the intensity of the light reflected under certain frequencies actually tells us what it looks like. So it doesn't really look like that, but that's the idea, is that there's something, there's a molecular process. That was kind of a crappy video, sorry. I like that website, but it didn't give me the right stuff for that particular case. So most of the data that is available to us, so there's a couple different places where you can look for data, and I'm being US-centric here. There's the European sources, which I'm not considering. Actually, the European sites actually have some better resources for proteins, actually. There are resources at the NIH for proteins, but generally I would probably go to the other, but I haven't given you that information. I'm going to focus in on GEO, which is the Gene Expression Omnibus. So this is the collection of everything gene expression. So for a long time, if you did published a gene expression study, you would be expected to put your data into this repository. A long time being 10 years or something like that. So I know that when my son was a baby, I went to a meeting where we talked about the data format that's used here. But I don't really know what happened after that, but I know some of it went into this. So there are multiple different things here. There's basically two different, I'm sorry, I didn't make that a link, but there's two different things. We have data sets and then we have profiles. The profiles will show you information about how the expression occurs. And the data set actually tells you about the data that we've got. And there's three, essentially we're sort of talking about a database here. We have a data set corresponds to a series, which is it's a study, or we consider it just a collection of samples. The sample then corresponds to whatever the sample is in the study. The example we're going to look at, these are things where the brave little mice sacrificed their lives for us have this data. So individual samples correspond to an individual mouse, and in this particular case, a particular region of an individual mouse's brain. So different studies will have different things. So a lot of the examples that we might see, so if we were looking at, in human cases, we could look at brain, but it would be mostly post-mortem brain, so it would be people who have died and donated their body. Their brains could then be used for collecting samples. It could be blood taken from the arm. It could be modified white blood cells, which is another common form of expression in humans. So there's like different kinds of things that show up. Sometimes there will be biopsies, like the kidney diseases that I've worked on. They actually took biopsies from the kidneys of the study participants, so we actually have the information from that. It could be muscle, tissue, it could be all kinds of things. But generally, with people, we don't like poking holes in them is somewhat dangerous, and poking a hole in your head is really ill-advised. The kidney's bad enough, actually. If it's not done correctly, you could bleed to death. So we have the series, the sample, that really describes what the data we're going to deal with. And then we have the platform, which is the technology. And I think there's something like 7 different manufacturers of microarray technology. Then there's the RefSeq is a different matter. But there's 2 main platforms that people use. One is called Affymetrix. Both of them are companies. Affymetrix and Illumina. So Illumina is the company that almost everybody's used, not everybody, but it's the high throughput sequencing technology. That's sort of the standard one that a lot of people use. And it's where the technologies are very similar for the microarray. The difference between the two, and I didn't make a slide on this, but the difference between the two is that Affymetrix, what they did is they actually represent a gene or a transcript of a gene by multiple probes, so multiple cDNAs. Some of them are a little bit off, so it's kind of this weird combination of mix of stuff. So sometimes people will filter those out. But you end up with a set of probes that correspond to a gene or a particular transcription of a gene. And then Illumina is just longer. They use a longer probe. They use a single long probe. And it might, if you remember the picture back with the central dogma, we had the red chunks that would fit together. What they're trying to do is come up with probes that would match up with these different ways of splicing those things together. So it actually, if you were to look at that probe across the whole normal sequence, it would be a little piece here and a little piece there, based on how the gene was in the genome, how it was split up between those introns and exons. Okay, so the probes are different, the different technologies are different, so we actually have to look at them in different ways. Unfortunately, what I did when I started looking for an example is I did not grab the raw data. So maybe that's positive for today, but the data that's in Geo they typically will provide the raw data, which is what comes off of the machine, or whatever the technology is. And they will provide usually the normalized, cleaned up version. So it's already tagged. So some of the process that we go through is we have to identify the probes that with a gene. So ordinarily, if we were to do a study, say we were to do a gene expression study, everybody, we took a swab and we did some gene expression based on what came out. We would have that particular, we would get the data in a particular form. It would be tagged to or annotated with the current version of the genome, and we would be looking at, everything that we got would be up to date and current. What happens is some of these data sets, they go back pretty far. The kidney data set that I worked with, it had samples taken over like 5 years or something. Some of them were on one chip. It used to be that was, in order to do the whole genome, you had to do 2 chips with the Affymetrix, and they decided to save money, they would only do one of them. All right, so originally. And then by the time they got to the end of the study, it was 1 chip. The whole thing was 1 chip. So we have different sets of probes based on which thing they came off of. And so you end up with this weird thing, and they're done in different batches, so there's all kinds of data cleaning that we have to do. But the key thing is that over time, so if, like I said, if we were to do something now, we'd get all up-to-date information. But the probes that were used in a study 5 years ago are based on the genome probably 7, maybe 10 years ago. And so, you remember what I said about genes packing up and moving? Well, we need to be aware of that. So what happens is that probes can actually no longer be valid. So we can have, or they might end up tagging multiple genes. So because of different ways that, and they might not be as specific as the designers thought they were. Yeah?
So with the 2 different technology types you stated, it seems that we're just measuring, or the data is just measuring if a particular variant of messenger RNA is there or not, and not the amount, like not the frequency.
Yeah, you're just counting the number. So the intensity of this, with the spotted microarray, we're using the intensity.
Oh, okay.
Yeah,
Well,
so that's a good question because I forgot.
Well, with arrays, you get relative abundance. So you compare like a case and a control.
Mm-hmm.
And then with the RNA-seq, like the next generation sequencing, you can actually get absolute.
Yeah, you
can
actually
actually
count them, right?
absolute abundances.
Yeah.
On that, RNA is substantially more unstable than DNA. And so if I draw my blood and I spin it down and let it sit for 10 seconds versus letting it sit for 10 minutes, the RNA levels are going to be different, even
Right.
though I'm the same blood file. Is there metadata on these samples? On
Yeah,
processing?
so we
or-
know some things. So it depends. Typically, if it's based on the tissue, there may be some metadata that tells you how long it. I mean, they'll tell you what the protocol was.
Okay.
So those sites, if we were to click into and look at a particular data set, that would. Now, it depends on the study how good a job they did in keeping track of that. With the brain samples, actually any tissue that's removed in a procedure or where there might be decay, then there's an issue with changes that occur because of that. The brain, for instance, typically with the brain you'll see metadata that says about pH, which is, yeah.
When you say the timeline of the data, was that from the same person?
So when I was talking about the timeline with the kidney study, it was that over time they were getting subjects, so it's like over 5 years new people were coming in. So they only did 1 biopsy per person, but the different people would be at different times. So if we wanted to study expression, we would pool everybody together and look at their data. But we have to deal with that. So I sort of left all that stuff off just by accident of what. I kind of figured we'd talk a lot about this stuff, and I've done enough of these that I know that it doesn't go as fast as you would think it would. So So Geo is a good resource, and actually there's a guy named Atul Butte. He used to be at Stanford. He's now I think he's running the precision medicine activities at UC San Francisco. Just moved there at the beginning of the month, and one of his research associates, now faculty at UCSF, started this project called StartGeo. And actually, so I should say what Atul's lab is known for, one of the things that they're known for is taking a lot of the open data, the public data, and even some of the stuff that we can't get too easily, and using it to combine it together in order to get results describing diseases in general. But one of the issues is that the data is not, if we actually look at the data that we're going to use, I actually had, in order to group it the way that we're going to use it, I actually had to drill into the data each individual thing, like there's 36 samples, and I don't know, 12 of them, the labeling doesn't tell you enough information. So you have to like drill into the sample information to find out which kind of mouse it is that you're looking at and where it came from. So the goal of Dexter, who is this, who has this project, StartGeo, so this is a live site. It's sort of flaky, but it's kind of interesting. He actually can do some of the things that we would do with the data, but they can do it. And actually on Geo, you can do some of the things we might do. Actually something similar to what we'll do with R. What he's interested in, so this is somewhat of an advertisement. If people are interested in working with this kind of data, what he's doing is actually setting it up so that these samples, like for instance the samples that we're dealing with today, I mean, he's looking at human data, but if they were included, they would be tagged with an actual, that have a tag that said this came from this kind of mouse, instead of being, you know, sample whatever it was, sample blah blah. And what he does is people who have biological background or biological knowledge, they go in and it's kind of a crowdsourced tagging. I've earned $45. But I haven't seen it. He told me I earned $45. But anyway, so if there are people who are interested, he would love to have conversations about what kind of APIs you want. And we'll see actually with the Geo, there is an R package that downloads them. So that's actually what I sent you. So it's the StartGeo site there. But anyway, if people are interested. Okay, getting closer to nitty-gritty here. So in R, R is pretty standard bioinformatics tool. So I've done a lot of bioinformatics related work, but I'm a computer scientist, and I usually tell the bioinformatics people what to do. And sometimes they listen to me. So my R is actually a little rusty, I realized, when I started working on this. But we've got enough to go through. So Bioconductor is kind of a deep collection of R packages that relate to biological data. The canonical way of installing is you use this BIOS Lite, Bioconductor Lite script with the source, and then you run it with no arguments, and then it installs Bioconductor into your R. Okay, so that's the first step. So I sent all this stuff in the message that when I was worried about data download. I'm not sure if it's really a problem or not, but we'll see. Okay, so I'll go back in. We'll talk a little bit more about setup here. But we're going to use this series. So this is a series identifier. So there's identifiers that they sort of indicate what they are. So GSE is the prefix for a series. I can't remember the other ones, but they show us the data. Actually, I want to show you one thing on that Star Geo site real quick, if I can get there. So see, it still has the Stanford on, even though we're. So one of the things that is interesting is about this is he does the he can do sort of default analysis based on the data. So I'm not really sure exactly what he's doing. So it's looking at basically using what genes are expressed and then looking at sort of a correlation across them. I'm not precisely sure. This dendrogram is showing you how similar things are. So things are, the tree will be, these things will be closer to each other. I couldn't tell you what PAC and ACC are, some sort of cancer. And then, so they're showing sort of how close the genes are. So he's already doing some of the processing that gives you sort of a broad understanding of how based on the data that he's got and the analysis that he's doing, how closely related things are to one another. And these are based on data sets, so multiple data sets. So you can see, in this case, there's, if I get my numbers in the right place, 41 series for that one. So that's 41 different data sets. So what he's doing is actually taking different studies and putting them together. All right, and that's kind of the goal, that the advantage of this kind of data for us, or for people that are interested in using open data to do something, is that these kinds of data sets make it possible for us to look at a lot of things, a lot more data than we could possibly collect on our own. All right, so there's other stuff there, but that was the point I knew I wanted to make but forgot. So let's look really quickly. I already talked about this example. This is a study. The reason that I know about this data set is actually I had some students working on a project for somebody that I've worked with who's interested in ataxia. Ataxia is a human condition where you basically lose motor control. And there's different kinds of ataxia. And it tends to be genetic. And in this case, there are different mouse strains which are modified. They're modified so that they behave in particular ways that kind of look like ataxia. So some of them are pretty awful, actually. So Stargazer, the mouse basically, it's a description of the phenotype, which is that the mouse looks up at the sky, and it's actually unable to eat. So they don't live very long. So basically they're born and then they don't procreate, so that's sort of a problem. Tottering would be, that's kind of its classic ataxic behavior, where they're sort of falling over and then lethargic, because the detail, I'm not sure if they don't just get up or what it is, but they're modified in ways that make them look like they have different symptoms of ataxia. So what was done in this case was that the expression, these mice were taken to a certain age, and then they were euthanized, and then their brains were sampled. Okay, so let's look at this particular example. I'm not going to go through it, but this is the kind of detail that we see in this data set. So this is, or this series. So this says where this thing's come from. And it's interesting that there is a publication to go with that data set, but it's not there. So it's the kind of thing you run into. This has a platform, so the information here, so there's information about the study. It has the platform. It has more. It actually has a project there. So we can click on that, but that's actually going to go to a general set of information. So that says we're dealing with a microarray, which is the Affymetrix mouse genome 430 version 2. And then it has 36 samples, and these are all the samples that we've got. So you can see I was clicking through here. Some of these are, you can see some of these have Stargazer, so Stargazer that has the tottering and the lethargic, but then some of them are labeled in different ways. Right. So the samples are not consistent. Then we have down at the bottom, actually we have this analyzed with Go2R, which we're not going to do because we're going to do something similar. That actually uses R online, so you can actually do some things quickly that way. And then there's different kinds of files that are there. So the one we're going to actually work with is this first one called the soft file. The one we probably want to deal with in the long run, if we're combining lots of things, is this this raw file down at the bottom. The cell is the for Affymetrix is the raw data type for the expression data. Okay, we can go into more detail there, but I just wanted to show you that. Okay. So, all right. So the setup we have, we're going to use. We're going to mainly use two libraries. Biobase is the kind of the base. It provides a lot of the utilities that you use. You can see these things on the documentation is available on Bioconductor, and the typical. You can get the typical R documentation for each one of them. The GeoQuery allows us to query the data set, to actually pull it down. And then the lemma is the statistical package that we're going to use. So our setup is we basically need to, we use BioSeedLite to install those things, and then, so without the arguments, we'll install Bioconductor. With the argument, we'll then, the vector of package names, it will download each one of them. And then, of course, we load the libraries before we start. Okay, so I was delayed in getting my act together, so I didn't get this up on the group site, but I have it on my own. So what is that? Probably not very useful. I don't know if can you go to my BJ Keller GitHub?
It may be private. We could fork it over to the Data Club.
I think it's pub. I set it up as public, so let's see.
Yeah, I got it.
Okay.
So go to your gists.
Am I going? Oh, BJ Keller gist.
Is it? Well, I'm on your main GitHub page.
Yeah. So if you go.
How do I get to the gists repositories?
So it's GitHub BJ Keller. Yeah. Yeah, so it's linked from the top of the page. So if you go to GitHub /BJ Keller, it'll get you there. All right. So it has the script. Some of this stuff I already sent you. And I've already done some of it in my, it's already downloaded the data, grabbed the data.
Ben, is it possible to make the text a little bit bigger?
Let's see. I'm not sure on the R, do you know? Format. Yeah. Okay, that would make sense. And all right, so from the gist, we have the stuff that I've already talked about. We love the libraries. We're going to use the data we're going to use is from this series that we talked about. We're going to use the getGeo function to grab it. And I haven't looked into the details of this because this is actually today was literally the first day that I've ever used this function. Usually when we do this analysis, you have the data sets. You already have the data sets and you're not grabbing them from the website or from the site. What this will do is it will get get the file. And unfortunately I ran through this. The data set that we get is not what we want. So let's look at what GSET is over here. So if we just look at, so after everything's loaded, if we just look at GSET, it is loaded as, the thing is, it's an expression set frame. So there's a frame with, I believe it's a frame with called an expression set, and it's got the, all of the data that we need in it. But we had to extract it from, I need to look at exactly what this is doing, but we had to extract this expression set out of the object that we got. So that's what this little business is about. And I put all of this stuff at the top. We don't actually use this information until later, but I put it at the top because I wanted to send it out to you just in case you could download it. So all of these steps will come, and this actually, I'm not really sure what the warning's about, but I think there's a download problem for this particular data set, or this particular part of the data set. So the 30 warnings are telling me that I couldn't get that GZIP file, but I already have it stored. So I get two things. I get the data, I get the actual series data, which includes the array information, the gene expression information, and then I'm going to get the platform annotation. And that says which of the probe sets correspond to what genes. So the probe sets in this case will tell you which probes are being matched up to the RNA. And so what the NCBI has done has provided their own annotation that attaches to that. So some probe sets don't actually match to anything because of changes. I'll talk a little bit about, if we've got time, I'll talk a little bit about the difference. If we were to start with the raw approach, we would actually change the probe sets that we were working with. But in this case, we use the default sets and then reinterpret. So this gives us the annotation, which basically tells us which genes we've got. We're going to muck with the labels a little bit. And then, you got it onto the page and you've got what you need? Yeah, okay. So, let's see, where am I?
Can you tell me again how to get to this page?
This one?
Yeah.
Okay, so if you go to, so it's easy for me. So if I say, because I always go to github.com, BJ Keller.
Yeah, I've got that.
All right, and then at the top, you see gist.
Oh,
And you may get something different because this is my account.
no, I don't see just.
You don't see gist?
If you just search for, this is how I got to it.
Brandon just posted on the
Oh,
Meetup.
okay.
Oh, the meetup?
This is
So
how
the
I got to
Meetup,
it.
you can find it.
Google that.
Oh, okay.
Yeah, I have the disadvantage. It always goes off the page. Actually, I meant to put the slides up when.
Page four. No, okay, not sure. Okay, got it. I think.
Yeah.
Ben, we have about 15, 20 minutes left.
Okay, all right. So we'll just go through real quick. What I've done actually is tagged, so 36 samples, and I've tagged based on which strain they come from. So are they tottering, lethargic, or stargazer? And then, so there's G0 through G2, and there's one for each there. So
So I ran your script. Is this
is it for-
detrimental? Closing unused connection 3, or is that just got a warning message?
Which one? On the, it's probably the geo not working.
Geoexample.r.
Yeah, it may be that, I'm not sure exactly all of the weird things that can happen when you talk to geo.
I'm just wondering if everything worked properly.
Yeah, it's maybe not. Okay, so the things that we have, so I want to walk through the parts of this. This next step, so I've set up the samples. What I want to do is I want to look at the, actually look at the data. It's very small. And so this is a box and whisker plot of the data set. As it is, it's normalized. Yeah, I believe this data is normalized. It's not, obviously not median normalized, but I don't know the details in this particular case. But this shows us the data set.
Looks like it's quantile normalized.
Quantile normalized, yeah. Probably. That's if I remember it right, that was what it was. The colors then correspond to the different, for the different samples in of the different strains. All right, the next thing that we do, Lemma expects log transform, so log2 transform, so we'll do that. We're going to set up the model. So there's a couple different steps, because what we're going to do is we're going to do the expression. So this model is dealing with the individual expression. So I'm not, with the time and my level of remembering on all of this, I'm not going to go through it. Send you to the Lemma documentation. So we do a fit. So the pieces here, we are setting up for this design here is the key piece that's telling them what our how to fit the model, or what model we want to fit to, and then this step is actually doing the fit. The Lemma primarily uses a t-test action. Although there are some choices. The next step is now we want to set up to do comparisons. So you can see it's got this make contrast. It does all the comparisons for the different groups, and it uses as levels the output of the design. And then it's doing a Bayesian fit, or I believe it's a naive Bayesian now. Okay, and so in the end, what we're getting is what we can do with Lemma results is that once we have the fit, we can ask for what they call a top table, and so when I extract that. Oh, wait. I had to do another step. So the data is not annotated the way we want it to be, so this is using that annotation. So let me re-annotate the data. That's what these steps are. So these are re-annotating the data, which genes are which. So that's what all this stuff is about here. The top table was just selecting the best results from that. And then we're going to pick out the columns that we want. So in this particular case, you might want to, if you're going to play with this, you could obviously drop that. Look at it. All right, so then I've got a commented out section to dump the file. And what this looks like is, so when you do the write CSV, oh, I did not want to do Excel. Oh well. Who knows what's going to open again. Okay. So we get, this first thing is the identifier for the probe set, which is not very useful. The second thing is the p-value, which I think we used FDR. I'm not sure what we did, to tell you the truth. I'd have to go back and look. And then, so the second p-value is the unadjusted p-value, and then this F statistic is based on the combination of the three columns. Now the other information tells us which genes there are, and so these are things. I actually would have preferred to get the ref SIGKDDs, but this is how the script is set up. So in this particular case, it's telling me a gene. I could take that to the NCBI site and look that up and get more information about it at this point. Okay, so, and you notice I've actually got 3 of them of these class 2 that are there. Now, what I didn't put this into the gist, but if I wanted to look at one of these things, sorry, this is not going to get bigger because I don't remember how to do that yet. Emacs. So I could do a bar plot just selecting on the row. So OEX is the version of the expression data that I got from. So this expressions is taken from that expression data set. It's extracting the expression data. So what I had done is I'd ordered it by our groups, and then what I'm doing is then looking at it based on looking at that particular probe set in what happened?
It went to a different window.
Did I lose my window?
Yeah, the behind.
Yeah.
There we go. So I haven't done any color or anything, but in this particular case, what we would have to work on this format a little bit. So this is what we were looking at was differential expression between the other 2 genes. So this is kind of like, what we saw was that in this case, I think this one is, I can't remember which group is which in the middle. It's probably, however that's set up, it's tottering. So this one is, roughly speaking, it's up in, so these are the non-transformed values. So it's it's active, the particular gene is active in the other 2 strains and down and tottering. And so we could look at each of the genes like that. So this opens up a lot of different possibilities. The only thing is that in this particular case, so since we're running out of time, in this particular case, we're working probably for the purposes of understanding disease in general. We probably wanted to start with the raw data set as opposed to the normalized data set, because then we could combine it with other things. One of the things I forgot to mention is that people use mice to reason about humans all the time, but translating across is kind of complicated. It's possible. It's possible to do, but genes are actually in different places. They have the same, sometimes they have genes that do the same things and they look very much alike, but they're actually in completely different locations in there. So it's not like, so you might have two genes that are in the human that are side by side, but they're on the same chromosome, but in the mouse they're on different chromosomes. So all kinds of weird things that happen when you're going across species like that. The other thing is that I grouped just based on the mouse strain. I'm probably actually interested in knowing about differences between, although I have really small numbers there, differences between the brain regions there. And so in that case, we have to go back and relabel by group. So that's one of the places where the tagging is useful. So as I said, I would prefer to work with the raw data, and actually it's different steps. This tutorial actually, I'll put these up where you can get to them and post the link. And this set of steps actually gets you to, or as described in this document, actually downloads the raw data. And you can do that with the geo package. And it basically download it into your directory. You can do that otherwise, but as part of your script, you can do that. The other thing, as I mentioned, the genome assemblies change, and so the probes change identity. So this is really a problem with Affymetrix. I haven't seen this done for Illumina, but we've done it ourselves, where basically what we do is we come up with our own chip description file, and it describes what probes belong to what genes. And so this is a site with people that I used to work with in Michigan, where they have this. They actually will periodically just churn through all the data, all the genomic data, and match it up to the probe sets, and then publish a new set. So they have multiple. If you were to go to that page, they actually have multiple different versions. And it's so useful, it's actually sucked into the Affymetrix package in Bioconductor. So there's a package called Affy for Affymetrix, and a package called Lumi, L-U-M-I, for Illumina. The Affymetrix one has some version of that set if you want of these things. And they have on that site, they tell you how to use them in your analysis. Okay. So just to wrap up, there's lots of different resources, lots of different sites that have a lot of data. So these were just things that I found useful over time. Of course, the NCBI site, which we were already using, but there's a lot of other data there. So if you click on that, there's a bar there's a menu on the left. It has resources and data, or data and resources. If you click on that, then it will show you all of the different kinds of data that they have. And it's a great resource if you want to learn about some biology. They have tutorials and all kinds of, they actually have some books online there. A lot of different things that are available. The ENCODE project deals with identifying information that tells you, help identify places where there's some sort of activity on the genome where it's opening up for instance, to start regulation, so for places where binding to the genome can occur to start transcription. So a lot of it, cool, good information there. That's a really new resource. Well, not really new. It's within the last 3 years. It was a really big deal when it came out. The genome browser, of course, that we looked at, it actually has this table viewer, which is useful for downloading information. So one of the a couple of the projects that I worked on, I was looking at those models of genes, and I wanted to come up with sort of the best. Actually, all I really cared about was where the ends might be, and so I used that to download all of the different models that are possible, and then come up with the one that was come up with the combination that was the longest. So I used that a lot. And then actually a really new resource, which I just put up there because it's kind of cool, is this. It's not even on the NCBI site. It's this import, which it has to do with immunology data sets. So this is a really new kind of thing, a new set of data that's sets of data that are available. Adaptive technologies. This is based on a different kind of technology than what adaptive technologies does, but it's sort of in the same kind of principle. So there's a lot of different data about immune response, and that's really interesting. A really completely different type of biology, and another possibility. So there's a lot of open data there. So these in particular are good places. This is, UCSC is one of a number of different places that's kind of aggregating data. So it's a good place to go find stuff all in one place. You could get all of that, but you would have to go shopping in multiple different locations. So. Okay, sorry, it was a little long and we're just perfect. Okay. Could have done more hands-on, but. So the kinds of things that we can do with this data, obviously, some of the interesting questions that we might ask about data is when things are working together. So a lot of work that's done is looking at, correlated expression across, in the StarGeo, I showed you the dendrogram, but it's based on correlated expression across different studies or different diseases, actually. But there's all kinds of things we can do. Some of my favorite analyses are involved using graphs to look at relationships among genes. All right, thank you.
Maybe another session for the hands-on stuff.
Yeah. Yeah, once I get it, actually, I was like going, oh, I want to do that, and it's like, no way I'm going to get that ready. So, yeah, there was a lot of, and I can pick something else out.